Hello, welcome to this UCL lunch hour lecture on genes, depression, and diversity. Can DNA unlock the biology of mental illness? My name is Andrew McQuillan, Professor of Molecular Psychiatry in the Division of Psychiatry here at UCL. It's my honor and pleasure to introduce today's speaker, Professor Karoline Kuchenbaecker. Karoline is Professor of Genetic Epidemiology at University College London, where she leads the Diversity in Genomics group. Her research focuses on the genetic and environmental risk factors for disease by leveraging unique characteristics of diverse populations. She has led the first globally and ancestrally diverse research studies of the genetics of depression and is the PI of the ERC-funded Diverge study, which has recruited over 13,000 participants with clinical major depressive disorder in Pakistan. She's developed methodological standards for diverse samples, as well as innovative methods to empower locus discovery and to assess the transferability of genetic risk factors. She's also scientific lead for the diverse data projects at Genomics England, where she's supporting programs to ensure equitable research and application for genomic medicine across minoritized communities in England. Karoline is co-director of the UCL Human Genomics Centre and co-founder of the London Genetics Network. I'm going to hand over to Karoline now for her talk.
Thank you so much, Andrew. It's a really great pleasure to be here and talk to you today about genes, depression, and diversity. And I'll try to tell you a little bit about whether DNA can help us unlock the biology of mental illness. There we go. So when I talk about my work, I sometimes get the question, well, oh, depression. So is this now considered like a real disease? And that's quite concerning that people ask that because, yes, depression is a real disease and it's actually a very severe disease for lots of people. Here, this is a study by the WHO, and they looked at the overall impact of different chronic conditions on health. And out of the ones they considered here, and a gigantic number of people, the one with the worst overall impact was actually depression. And that's not the only thing. Depression actually very often coincides with other conditions. For example, someone might have type 2 diabetes, but also depression. And the combination of conditions like that seems to be particularly bad. For major depression, very often the impact is not only actually with the affected individual, but also the people around that person. For example, this study demonstrated that women who suffered from depression before they got pregnant actually had a higher risk of negative birth outcomes for their babies, for example, lower birth weight or higher risk of preterm birth. Now, I know that this can actually be quite scary for people, many of us who do suffer from depression, to think about the impact. And there is, of course, no blame on the individual. But it is key to understand that this is a really severe disease with very far-reaching impact. It's impossible to really give people an idea what depression feels like. And I think those who haven't been affected sometimes think it's very similar to low mood, but it's not quite like that. And here are some quotes from, really influential sources, let's say. One is a book by Andrew Solomon where he said, "I could not bear to think, to look, to experience. I was desperate to be nowhere at all." And Winston Churchill, also is quite well known now that he also suffered from depression, said, "I don't like to stand by the side of a ship and look down into the water. A second's action would end everything. A few drops of desperation." So the almost seduction into, ending it all. But I also asked generative AI, so ChatGPT in this instant, and um, the app was very eager to tell me what depression feels like. It said, imagine being locked in a room with no windows, no doors, and no way out. You scream for help, but no one hears you. That's what depression feels like. So I think part of the challenge with people, some people not knowing what severe disease depression actually is that we don't have such a firm grasp on the biology behind all of this. So for example, for type 2 diabetes, today we have a really clear model of what actually happens, what goes wrong, what cells are involved, and what's a sort of cascade of action. And that can also help us develop therapies. For depression, it's been a little bit more challenging. We assume it's been something to do with the brain, but what I'm going to talk about is exactly how we can actually get to a much better biological model of the disease. And what has made a huge difference here is our knowledge of the human genome. So the human genome is gigantic. It's a set of extremely ridiculously long molecules, and it's often and quite helpfully represented by this long list of letters here. And you can see them actually printed in books and books at the welcome just behind UCL. There's also this YouTube link, which will show you for a very long time this long sequence of letters, just to give you an idea really how long this molecule is. And we actually have many, many copies. So it took a long time to actually read this all. It was a gigantic effort. We've obviously known about the existence of DNA for quite a while, but it took until 2003 for people to actually publish essentially the full book of letters. That's what it is. It was also extremely expensive to do this. But knowing this sequence as a sort of reference starting point has actually started off a huge amount of research and enable us to look into the genomes of many people. The challenge now is that it's so big. So here, this is 3 billion bases, so essentially 3 billion letters we're looking at. And it does encode 22,000 different genes that then go on to make proteins. I'm going to avoid using, too much scientific genomics language, but for anyone watching this on YouTube later on who can actually pause in case you're interested, I did include a bit of a genetics glossary here. But one term I do want to briefly introduce because I will be using it a lot, and that's genetic variant. So imagine this long sequence of letters, and then also keeping in mind we got one copy from mom, one copy from dad. So we have 2 copies, and all people at most of these points in the genome are actually the same. So as you can see here, the first point, everyone here, for example, has this A. And that's not very interesting for research. If we're trying to understand, for example, causes for disease, we're looking for something that is maybe a little different in people with depression compared to people without. So the points we're really interested in is where some people may differ. And that you can see here, for example, this person at this point has an A, but the other person has Gs. And that's what we call a genetic variant. So a point somewhere in the genome where 2 people have a different letter, essentially. And there are other words for it that actually mean something very similar, for example, a different allele, or a particular type of variant really is this single nucleotide polymorphism. So what does genetics actually have to do with depression? There's really clear evidence that it has a lot to do with depression. In fact, depression is partly a genetic disease. How do we know this? There's a large amount of studies, but a very good way to look at this here is to use information from twins. For example, this study here looked at data from 50,000 Swedish twin pairs, and monozygotic twins are genetically basically identical. So the idea is if one of the twins develop depression, then we can look at whether the second twin is also more likely to get depression. And if that's the case, more often than you would expect, you can see that there is a genetic basis, heritable basis for the disease. And you can also estimate roughly how heritable depression is. And here they estimated that it's about 40%. So 40% of why people get depression can be linked to genetic factors. The first bit of research on depression genetics I do want to mention is what is easily the most famous depression genetic study in the world. It's actually probably the most famous genetic study in the world. It's very commonly cited. It's been covered by the media a lot. It has quite a complicated name. It's the serotonin transporter interaction hypothesis that was tested by this paper. And the background is that, so serotonin is a little substance in the brain that helps brain cells communicate. And it's also the target for antidepressants, medication, especially the most common medication prescribed here in the UK for depression. They are primarily targeting serotonin. In this study, they looked at a particular genetic variant. And this genetic variant, what it does is impact a gene that's encoding the serotonin transporter. And essentially the idea is the variant influences how much of the transporter is made and possibly how much serotonin is available in the brain. So they looked at this genetic variant in about 1,000 children, and they also looked at whether they had depressive symptoms or not. If they just looked at the gene variant and who had what version of it, it actually didn't impact much on the level of depressive symptoms. But then they also accounted for whether, which of these kids were actually exposed to stressful things. And here on this plot, they counted how many stressful events each kid was exposed to. And that's a very strong predictor for depressive symptoms in general. What they found here is looking at the different versions of this genetic variant is that the kids with one of the versions, LL here, even if they were exposed to quite a few stressful life events, they really didn't show much of an increase in depressive symptoms. They were almost protected. But the kids in the top line here who had what they thought the more unfavorable version of this genetic variant, if they were exposed to stressful life events, their risk of depression shot up. And that's what we call an interaction between the environment, stress, and a gene variant here. It really got a lot of attention, and this research was published in possibly the most prestigious scientific journal called Science. It was also the first really big discovery in this area overall at a very, very early time. You could see 2003, that's just about when the full sequence of the human genome was published. Now, fast forward a number of years, and actually today, what I really want to share with you is that this hypothesis has actually been disproven. So unfortunately, this finding did not replicate, and there's been a lot of additional work to try and check if people could find the same thing in different studies. People even reanalyzed the same data set and they just didn't find the same effect. This has had a very significant impact on research in this area overall. Yeah, because we're all a bit like burnt children. There has been this very influential study, and today, when you look around the internet, actually, there's still large parts, or, even amongst scientists who maybe not directly in this field, who are not aware that this has been disproven. That's how far it went. And then actually there was much less coverage of the fact that, evidence is actually not supporting this anymore. So what has happened is today we're extremely careful, we're methodologically very, very rigorous. And we've also changed the way we're starting these studies, as I will tell you in a second. So in this early study, they had a hypothesis about a specific gene and region that might do something. I mean, serotonin just sounded so good, right? It sounded like it should be serotonin. But with a disease where we really don't know that much about its biology, probably the hypotheses we can form are not always very good. So it's turned out that a better way to do this type of research is to go into it without a clear hypothesis and just look at as much of the genome as you possibly can. And here is an amazing technology that has actually made it possible to do this at very large scale. So we can use these little things here. They're called DNA microarrays. They're for reading DNA. They're fairly affordable. They're quite cheap. So you can use them in thousands of people. And they cover a good proportion of the genome, but importantly, not everything. So it's a good cost-benefit ratio. And what these little things enable us to do is what we call genome-wide association study. So we're not just looking at a specific gene anymore. We're trying to cover as much of it as possible. And then we see what we find rather than being driven by a very specific idea about what part of the genome might be linked to depression. So then what we do is we invite a number of people with depression, let's say people here in pink, and a number of people without depression, who we tend to call the controls, into our study. We look at the different genetic variants they have. And then basically what we're doing is we're counting how many people have a certain version of a genetic variant. Let's say this A version here. And then we're just comparing, it's actually quite simple, between the cases and controls. And if the cases have this A version more frequently, as in this example here, we are kind of then assuming that the A version of this variant, or this variant per se, may actually be linked to depression. And this variant, or something in this region of the genome at least, may have actually contributed to the cases developing the disease. When we do this at scale, for example, at the moment we're about to finish recruiting people into a very large depression genetic study in Pakistan, we first do data collection. So we plan the study, we get ethics approval. And then in this case, we go into different clinics, we're working with psychiatrists, and we're asking depression patients if they're interested in participating in the study. If they are, we ask them loads of questions. And we do a blood draw. So we take a little bit of blood. Other studies sometimes actually take saliva, which is a little bit more convenient for people. And then in this case, the blood sample is shipped to UCL Genomics, which is not very far from me right now. And at UCL Genomics, there's this amazing lab where they take the blood, they pull the right cells out of it, they look at the DNA, and they use these DNA microarrays to genotype people. And then magically, this somehow appears on our computers with yeah, a lot of advanced technology behind it. Then, not least driven by the serotonin study I mentioned, we do extensive amounts of quality control. So for example, for the Pakistan study, it's not even finished, but we've already spent a year working on our quality control of all the data we're getting in. Then we do a bit that's called genotype imputation. It's not so important for us here. And finally, we're doing the analysis, which is basically what I just showed, looking at whether the depression cases have certain genetic variants, a version of it more frequently than the controls. And this is the sort of primary main thing people get out of a GWAS. It's sort of the result as in sort of one image. So what this shows is each of the genetic variants we looked at and the result for that variant. And the key result we look at first at least, is a statistical thing called a p-value. And it tells us how likely it is that this variant is more frequent, say, in the cases. Ideally, we want a very powerful p-value, and that they are shown here as points that are really high up. So a point that is high up in this plot represents a genetic variant that is linked to depression. And you can see the variants are actually ordered by their location in the DNA. So number 1 down here is chromosome number 1, and then we go from the first position on chromosome 1 to the last. Now, again, we're being extremely rigorous. So in order to cause something significant, which is what we're looking for, so we want to be quite confident that this is not just a chance finding, we're looking at this line up here, and only genetic variants that cross this line we're going to investigate further, and we're quite confident about it being linked to depression. So this specific plot is actually one of the first GWAS, genome-wide association studies of depression, and it was published now about 15 years ago. So as you can see, actually, none of the points cross this line. So there wasn't actually any specific finding in terms of gene discoveries or so. This is another early study, less colorful, but as you can see again, none of the points crossed the line. So at that point, researchers, got together and said, okay, let's collaborate. Maybe we just don't have enough data. So they published what they called a meager analysis of genome-wide association studies. And nothing crossed the line. And that's been really quite frustrating. And there's been whole research papers written just about, why this might be the case. Because at that point, now about 10 years ago, lots of other diseases, they actually did have discoveries in terms of, using similar technologies. So it did take a little more than this to change that. And the successful strategy in the end was, to go really big. And really big in the, one of the first successful studies meant 130,000 people with depression and a huge number of controls. And that was only made possible because of researchers getting together in the form of a big consortium called the Psychiatric Genomics Consortium and sharing all of their data. That's made all the difference. And for genetics overall, that's been the success strategy in many cases. So this is a gigantic consortium with, hundreds of different researchers from around the world. And the data that's now been shared there is, we're talking millions of people. And this is obviously not just about depression. This consortium also focuses on several other psychiatric outcomes. So this is the plot for this first really successful study, and it picked up 44 different genetic variants linked to depression. Very quickly after, things went even bigger, and 100 different variants were discovered. And so this year, with this Psychiatric Genomics Consortium, we published the largest study today in a big, giant collaborative effort involving loads of researchers. We had almost 700,000 people with depression included in this research and 4 million controls. And we discovered 700 different genetic variants linked to depression. So what I'm aiming to do next is talk a little bit about what we've learned from this absolute breakthrough and discovery that took such a long time to get to this point. So one of the first things researchers noticed early on is when you zoom into this, it's literally the same plot as before. It's zoomed into one specific region that crosses the line, and then you can look at where it is more precisely in the genome, and you can overlay here where the genes are relative to this variant. And one thing that emerged for many of these different variants, the vast majority, is that they don't actually change the coding bits of genes. Most of them are actually completely outside of genes. Like you can see this one, it's near genes, but it isn't located in a gene. That was surprising. So, so what do we know about the genome outside of genes? Well, it's been called junk DNA for a long time. And people actually asked, why is so much of the DNA junk DNA? Because only a tiny proportion of the DNA does encode genes. And we know that quite confidently because that's been the big discoveries. Like we can read the coding for genes. There is literally like a computer code behind it, and we know we can translate it into proteins. But that's something like 2% or so. I mean, so it's like 98% junk DNA. Again, it needed extensive, collaborative, and really expensive modern research to understand that, well, it seems like all this junk is not really junk. And if you think about it, all makes sense. So all our cells have literally the same basic DNA, but they function very, very differently. And so in addition to having the code for a protein, a cell needs to know how much of a protein to make, and it depends on, what that cell's identity is. And also the developmental stage. So, different proteins are needed for a very early baby compared to, maybe an older person. And all this complexity is regulated in at least parts of this big junk DNA, and probably quite a big proportion. So what we're calling this is gene regulation. It's not changing the protein itself, but it does tell cells how much of the protein to make. And yeah, there's loads of complexity to it, and understanding which part of the genome do what is still an ongoing effort, and it's really important in order for us to understand the findings from those genome-wide association studies for depression and also for other outcomes. So it's not straightforward, and one of the biggest challenges for researchers like myself now is to go from the gene variant to the actual gene. But people have come up with loads of really cool techniques in order to do this, because the gene regulation bit is just a bit more tricky compared to having a variant in a gene that changes protein, instantly what the protein is. So we needed to do put in a bit of work in order to pin the variant to the gene it's actually regulating. But there are quite a few where we're now really confident that we know what is the actual gene. And I wanted to give you a couple of examples because this is where we're really learning stuff about depression. So one of these genetic variants is linked to a gene called NEGR1. And this variant has actually come up across several different ones of these depression genetic studies. NEGR1 stands for neuronal growth regulator 1. And it's very important for cells in the brain. So these neurons, the key thing for them is to connect in different ways. And it's about the connection that determines a lot of their functioning. So NEGR1 there's this little protein that sits on top of these neurons and help them regulate how they kind of connect to each other and communicate with each other. In mice, you can actually do experiments where you can see what happens if NEGR1 isn't functioning anymore. And what we see in mice is that the shape of the brain literally changes. So for example, an area called hippocampus is notably reduced in brain volume in these mice where NEGR1 has been knocked out. And these mice also show abnormalities in social behavior, and there are particular tests that show depression-like features as well. So very strong evidence across the board that biologically, but also in terms of outcomes, makes sense in linking this gene to depression. So how might this work? Well, I think it could be a bit like faulty wiring. So this gene affects how neurons connect and communicate. And if this is out of balance, then the brain circuits that regulate mood and emotion may also be out of balance. The other part where it's very likely that this gene plays a key role is brain plasticity, sorry. Brain plasticity is changes that happen all the time where the brain needs to adapt. And this seems to be an important protein if you think about it, different stages, different ages and so on, where rewiring might be necessary, for example. So if the brain isn't able to adapt quickly, it may actually again get out of balance, and that might increase someone's risk of developing depression. Another key gene that has come out of this depression study, and actually also other genetic studies for psychiatric outcomes, is called DRD2. And that stands for dopamine D2 receptor. Now, dopamine is the feel-good neurotransmitter. So it's, again, a little substance in the brain that helps neurons connect, and it plays a key role for things like reward, pleasure, motivation, mood regulation. And that already sounds like it would easily have something to do with depression, doesn't it? So this receptor bit is something that is the key, but the lock to the key, if you think of dopamine as a key, when it kind of communicates across cells. And then the receptor is where the key locks in and transmit that message, basically. So again, there's been additional research done in mice, and here they used an even more advanced technology where they don't just knock out a gene entirely, they have a method to turn it on and off in the same mouse. And they realized that when they turned this gene on, they found reduced depression-like behavior in the mice. And if they turn it off, then the behaviors got worse. So very clearly, dopamine neurons really directly influence mood and also resilience to stress, they found. So here, this receptor might act as a gatekeeper. So if that's not working well, it can throw off the brain's reward and mood systems, and that again might make a person more susceptible to depression. It also has interesting implications for medicines. So for example, antidepressants, there's excellent evidence that they work. There's been extensive research over the years. But the question is how, and that's actually much less clear. So as I mentioned, the most commonly prescribed antidepressants work on the serotonin system. Again, that's a little a molecule in the brain that actually transmits information. And the idea was that maybe if this drug is actually helping you have more serotonin, people get better, and serotonin is involved in mood, then maybe the cause for depression is really that people with depression have too low levels of serotonin. But more recently, this has really come under attack, this hypothesis, and there have been doubts as whether this is really the main biological cause of depression. Most likely it really isn't. It seems to be a little bit more complex than that. And I would say the genetics findings I just presented are in favor of this. So for example, seeing things like this dopamine receptor coming up very strongly implies that it's not just about serotonin. There's other types of neurons and their connections that are involved as well. And there's even some research suggesting that these antidepressants that people use very commonly, they're not only working on the serotonin system, they may actually also impact other neurotransmitters such as dopamine. There's more we can learn from genome-wide association studies. And one other nice feature is that now we have this list of genes. And then what we can do is we can infer relevant brain regions from these genes because we know where these specific genes are actually read in what type of cells. Again, all of this functional research in cells has made that possible. So here on the left is a figure from an earlier study, which I think very nicely highlighted the different brain regions involved in depression. And on the right is some findings with very, very specific types of neurons that are implied. So it's not, it goes beyond a broad region from our latest work. And I won't go into too much detail, but for people who have been doing research in depression, they will find that there's a maybe non-surprising, really good match about the areas that from other types of research, like imaging studies, where people have technology to take pictures of different parts of the brain. So it's similar parts that are implicated here in the genetics research and areas that we already previously thought might be really relevant for depression. Very importantly, so yeah, we do want to understand the biology of depression, but ideally we also want real patient benefits. And so one of the things we're getting really excited about is the potential to point towards proteins or genes that encode proteins that could actually be the target of new drugs. So most drugs people take for whatever conditions, they target proteins. And if we know a new gene linked to depression, maybe that would also be a good new target for a new drug. And there's some research outside of what I'm able to cover here that suggests that these types of genetic studies are really useful pointers, for example, for drug companies, and make it much more likely that a drug will actually go through if there's also genetic support for a particular protein. And here we certainly found some really interesting ones where there's maybe already a specific drug, and it could just be repurposed for major depression. Now, another area people have been really hopeful about is using genetics in order to be able to identify people who are at high risk of developing depression before they even get it. And then, of course, you could try and, help them, support them, and ideally prevent that they ever get depression. So what we've seen here is that it's not one gene or one variant that's involved in depression. It's hundreds, and probably there's even more that we haven't discovered. So we're talking thousands here across the genome. And none of them is the main one. If you have it, you definitely get depression. It's not working like that. It's more like if you have a particular version of a gene variant, you're at slightly more risk. And then basically, if you have really quite a few of the unfavorable versions, together, we expect that they may make a bit of a difference, and those people are at higher risk. So we have a technique called polygenic risk scores that are essentially just counting how many of the bad variants a person has. So if you look at individual 1, they have 1 copy of the unfavorable allele for these different genetic variants. And so their total score in this example might be 6. There might be some people at the other end who have none, and so their score is 0. And then what we've been testing is, does the person with a score of 6 actually, or, do people with a score of 6, do many of them have depression? And then we'd expect that people with a score of 0, very few of them might get depression. So let's see if this is working. Well, yes and no. So this is showing essentially a version of someone's risk depending on where they are on the score. So the people on the side of the 10 here, they have a very high score. And are they at higher risk? Well, the point is higher up. So the answer is yes, they are at higher risk. But as researchers, we've looked at how much higher is their risk. And is this really good enough to say, well, most people with a high score will develop depression? No, absolutely not. So there's still many people with a high score who will never develop depression. And there's also some people with a really low score who will end up developing depression. So at the moment, this is definitely at a point where this is not really useful for personal prediction. So I would not personally look at my own score and hope that it will tell me if I'm at risk of depression or not. It's just not working well enough. For the last very quick few slides, I wanted to mention a big problem in the field of genetic studies overall, and that's diversity. Diversity in terms of global representation, but also representation of people of different ethnicity. And here this plot shows different, actually, genetic studies for different disorders. MD is depression, major depression. For example, schizophrenia, SCZ. And most of it is blue, meaning most of the studies had people who were of European ancestry. And for some of the disorders, at least at the point when when we published this review paper, there's actually been almost only people with European ancestry. So no diversity at all. And that is a big problem. So a key focus for my research here at UCL and my group is to extend this to ancestrally diverse groups. And we published the first genome-wide association study of depression genetics ever that really tried to be as diverse as possible. And I just briefly want to highlight that there's things you only find when you increase diversity. This is a variant we did miss in people of European ancestry. And biologically, it's one of my absolute favorites, the melanin concentrating receptor hormone. We also looked at the polygenic risk scores again. And what this plot here is really showing is that we know that there is a link between the score and risk of depression, but it's notably weaker if you take a score that's been trained in this big GWAS studies for people of European ancestry, and then you're trying to predict into people for another ancestry group. And that's quite risky because so eventually, if there is a use of genetic risk prediction, it wouldn't be very fair to have these scores that work much better in one particular group compared to others. So we and others will continue to do research that increase diversity. And we're also thinking about, in collaboration with leading global researchers, for example, in this paper here, how this can be done best, can be done fairly, and can really help build capacity in a sustainable way. So last but not least, I just wanted to mention our fantastic study here in Pakistan, our great collaborators. The initiative is called PARC, and we're really excited about that. And hopefully you'll hear loads more about research we do in this fantastic data set. And there is a gigantic number of people to acknowledge. These are all the authors for this big, recently published Depression Genetics GWAS. And these are key people who contributed to the work I've been showing here, in particular Shangri Meng, Georgina Navali, and Olga Yanakopoulou. So sorry for talking a bit longer than I intended to. But yeah, thank you very much.
Great. Thank you, Carolina. That's an excellent talk. So we've got a couple of questions on Slido, but if anybody else would like to ask further questions, please do go ahead and ask on Slido. There was a link to remind you. There was a link sent with your Eventbrite invitation to the Slido website. So we've got so the first question that came in was you mentioned Swedish and New Zealand-based studies. Are there any UK-based studies?
Well, there's plenty of fantastic UK-based studies also on these specific topics. So I mentioned this Swedish study because they have very good register information. So they have data on almost everybody to some extent, and there's this gigantic twin register. But for example, in the UK, an excellent twin study it's called TEDS, and there's amazing research, especially around mental health, that's been done in this study. Similarly, actually, the first depression GWAS one of the first I showed was done by colleagues at King's College London, and that's been another UK-based studies. Colleagues here at UCL in the Division of Psychiatry, they have been recruiting patients with depression, schizophrenia, autism. Andrew has also contributed a lot to this, and actually is recruiting today as well. So there's been a major contribution of UK-based data, dedicated studies, general population cohorts to this area of research.
Great, thank you. And obviously the UK Biobank has contributed data to the big studies as well. So how would you go about increasing diversity in research?
It's turned out to be much more of a challenge than what myself and others expected early on. So the field started out focusing on this particular one group, people of European ancestry. In the early days, when the argument was, oh, if we're too diverse, it might make it more tricky to have reliable findings. Let's start with this group and build on that. And then the build on this part didn't really happen because things were so much easier focusing on this ancestry group, and there were lots of resources for it. So it became essentially a vicious circle. But it's already, years ago, people have pointed out and published very prominently that this is a big problem in many ways. We're losing insights that we could have gained if we have more diverse global participants in our studies. And also it could lead to essentially unfair outcomes of these studies if we developing medicines or trying to predict risk. So I expected after these publications that by itself things would diversify. And what we're seeing year after year is that's not just happening by itself. So we need to make more of an effort. And that includes lobbying, talking to funders, and also being critical of maybe research or questioning research if it's done only in one ancestry group, and asking ourselves and colleagues if this is really the best way, if there were alternatives. And then collaboration is key, especially for global studies. So I would never be able to do and wouldn't be the right person on my own to lead research in Pakistan, obviously. So we need collaborations with these amazing researchers and ideally help support them build capacity, get their own research funding to do large studies. And that is happening in parts of the world. But it's still a long way to go.
Okay, can I ask you a follow-up question about the diversity? Do you expect that genetic studies in diverse populations could help to discover completely new biological pathways related to depression?
That is likely because the underlying biology for diseases, in the end, fundamental biology is shared across everybody. I mean, that's important to get it out there. We're all working in the same way, just because you live in a different country or so. But there are slight changes across the world about how frequent certain of these variants are or not. And sometimes it's just, it's been kind of random what variant are present in a particular group. And if we only do research in this group, so by now, what can be found in people of European ancestry in depression genetics has pretty much been found. If we go beyond this many, many variants, the contribution of each one is going to be minute. But there'll be other important variants in other ancestry groups where they're just more common, and we're missing them if we're not including people from this group. And that's been demonstrated in a number of studies for not just for depression genetics, for loads of different outcomes. So I don't expect to discover fundamentally different biology, but it's been shown that there is a bit of chance involved in what you can find and what you cannot. So let's maximize our chances of finding all of the core genes, and we can do that by being more diverse.
Right, thank you. And then to what extent do you think the latest GWAS findings provide evidence for gene-environment interaction in depression risk?
Oh, very, very good question. So there has been some work around this, not just this early study that I mentioned that, obviously doesn't have really evidence supporting it, but there is evidence that there are gene-environment interaction with depression. What we're struggling with is to pinpoint the specific genes and specific parts of the environment. And that is because the search space is so large. One of the things that are supporting this, so there's one set of studies, what they did is look at people who had some form of trauma or really stressful life event. And we do know about depression that is one of the core predictors of some, people developing major depression later on, and other conditions as well, actually. So they looked at genetics in people who unfortunately have been exposed to trauma or more severe forms of stress, and people who haven't. And they found that there's evidence that the genetics are slightly different, and that is a form of gene-environment interaction. Equally, we're seeing when we do this research in people from different environments, different countries, for example, we're sometimes not picking up the same gene variant that was found before in European ancestry. And one good explanation for this is a gene-environment interaction. So they're very likely to play a very important role, but they're notoriously difficult to pinpoint and find, which specific ones are important.
And a slight follow-up to that is do you think that using a project risk score study, for example, that examined the number of traumatic life events would be a useful additional research avenue.
So you mean polygenic risk score and the number of traumatic life events
Yeah,
together?
yeah.
Yeah, definitely. And that is something people are looking at. So, both for interactions, but also for prediction. Overall, for other conditions, people have found that even if genetics by themselves, they're not extremely predictive, but sometimes they help when you add them to a model that has other useful predictors of a condition. In terms of prediction power of genetics for depression, they're really quite weak at the moment. And given that we've already had so many participants in these studies, personally, I don't envisage us using genetics to predict depression risk anytime soon. But that's different for other conditions, just to, explicitly say that as well. Depression is probably particularly complex.
So another question is, does impaired brain plasticity contribute to depression or symptoms of depression? And could enhancing plasticity through interventions help counteract that effect? Well, it's not really your area, but
Yeah, but it's a hypothesis that's extremely popular right now, let's say. And I would say some of the genes that we're finding in genetics, they are supportive of this. They're definitely playing a key role for brain plasticity. So I am not the person to have the final word on this, but I can say there's supportive evidence coming from genetic studies. Whether I would opt for an intervention right now to increase brain plasticity, well, the challenge is not very easy to do that. So yeah, it's a tricky question.
okay, we'll get back onto your sort of area now. So there's two questions linked to sort of diversity and depression. So one is it possible for there to be a link to depression and ancestral diversity?
Um, yeah, interesting question. In a way, sort of, does your ancestry or diversity play a role in depression is the question. So um, I think there's no genetic evidence, and it's not a good genetic question to see if genetic ancestry causes depression. In a way, that's something we can't really tell because there are loads of non-genetic parts that change depending on your genetic ancestry. So because your genetic ancestry also links to what you identify as your ethnicity, basically. And people of different ethnicity are exposed to different things. They have higher, lower risk of diseases, but they also, experience the healthcare system differently. There's racism at play. There's, slight differences probably across the lifespan, and they're really tricky and hard to measure. And the genetics obviously relate to this. So there are different people of different ethnicity have different risks of depression. There may also be differences in reporting depression because this is not a disease that's easy to diagnose other than talking to people, and people may, explain things differently. And all of this makes it very complicated to compare across ancestries. So I would strongly suspect that any differences we do observe are more related to these small, complex environmental aspects that impact people of different ethnicity differently.
Great. Okay, so we've got I need a very short answer for a very complex question, which is, should we prioritize improving diversity in GWAS before introducing polygenic risk scores into clinical care? Or can we advance both without reinforcing inequities?
Great question. So this is my personal position. I do research on diversity in genomics, and I feel like that equity is extremely important, and it's not okay to roll out a technology where we knowingly had a massive data bias, and now we're rolling it out knowing that it won't be very equitable if we do. So my personal opinion is we first need to improve diversity before we should, roll this out into healthcare.
Great. Well, thank you very much, Carolina. That's fantastic. That's all the questions, which is brilliant. We've come to 2 o'clock now, and so I'd like to thank the audience for joining us. We had what 73 live streamers on YouTube, plus whoever joined us on the webinar, which is fantastic. And so the next lunch hour lecture is on the 18th of March. It's the title is Lawyers and Acting in the Best Interests of Clients. So that sounds fascinating as well. So thank you very much, Carolina, and thank
Thank
you
you
everyone
so
for joining.
much.
Thanks. Bye-bye.